You hatch from a small yellow egg attached to the underside of a leaf somewhere in a temperate garden in northern Europe. The egg is one of roughly 40 in a small cluster, laid in a neat grid by your mother who is already gone. She will never meet you. She laid the eggs near an aphid colony on purpose. She could smell the honeydew from several meters away, so that when you hatch, your first meal is already within reach. You are a larva. You look nothing like what you will become. You are elongated, dark, spiky, and alarming looking in a way that suggests poison even when you are not yet carrying any. You are approximately 2 millimeters long. The aphids around you are almost the same size. You eat one. The aphid does not go easily. It kicks. It releases alarm pheromones that signal the other aphids to drop from the leaf. Some of them do. You hold on and eat. It takes several minutes. Your first meal is complete and you are still hungry. Over the next several days, you eat constantly. A ladybug larva in its early stages can eat 20 to 30 aphids per day. You eat, you grow, you shed your skin as your body expands beyond what the previous skin could contain. You molt 4 times in total, across 3 to 4 weeks. Each instar, the stage between molts, you are slightly larger, slightly faster, slightly more capable of catching prey that tries to escape. The aphids on your leaf are not a stable population. Some colonies can produce 50 new aphids per day through asexual reproduction during the summer months. But you are also consuming them, and so is competition. Other predators work the same plant. Parasitic wasps that lay eggs inside aphid bodies, hoverfly larvae, lacewing larvae. You are not the only thing eating on this plant. You have to eat faster than the colony can reproduce or move to another colony, or starve. You do not starve. You eat 47 aphids on the best day of your larval life, and you move between 3 separate aphid colonies on the same rosebush over 2 weeks. Then something changes. Your body chemistry shifts. You stop eating. You find a leaf stem and grip it with your back legs, and you hang there, and your skin hardens around you. You are entering pupation. The transformation inside the pupal case is one of the more dramatic processes in insect biology. The larval body largely dissolves into a soup of undifferentiated cells, and those cells are reorganized according to a different set of instructions into something entirely different. Wings, a round dome of elytra, 6 legs designed for walking rather than gripping prey, a different brain in the sense that it is organized for a different life. You emerge from the pupa in approximately 2 weeks. You are soft and pale at first. The red pigment in your elytra takes time to develop, and the 7 black spots that will eventually mark your wings take several hours to appear. You are a 7-spot ladybug, Coccinella septempunctata. While the pigment develops, you are almost unrecognizable as what you are. The red and black pattern you are developing is not decoration. It is a warning. You produce a toxic, bitter-tasting alkaloid from glands at your leg joints, a chemical called adaline, among others, that tastes foul to most predators. A bird that eats a ladybug once generally does not eat one again. The bright coloration is the signal. I taste bad. Do not eat me. The system works well enough that many harmless insects have evolved to mimic ladybug coloration without producing the toxin themselves, which tells you something about how effective the warning is. But the system is not perfect. Young birds who have not learned the association yet will eat you. Spiders do not have the kind of memory that allows them to learn the association. They will eat you if they catch you. Certain parasitic wasps lay eggs inside adult ladybugs the way other wasps lay eggs inside aphids. The wasp larva consumes the ladybug from the inside, while the ladybug continues to live and even moves slightly during this process, still trying to function while something eats it. Skip forward. It is autumn. You have spent the summer eating aphids, occasionally mating, occasionally being confused with a harmless mimic by a spider that did not receive the foul taste message you were supposedly sending. You have done well. You weigh slightly more than you did in September, and your fat reserves are built up. You need them. You are about to hibernate. Ladybugs in northern Europe do not migrate the way monarch butterflies do. They aggregate. In autumn, adults from a wide area converge on specific overwintering sites, south-facing walls, rock piles, under bark, and leaf litter, drawn by aggregation pheromones and by the physical characteristics of the site. A single overwintering site might contain thousands of ladybugs packed together for warmth. The aggregation has a problem. A highly specialized fungal pathogen called Hesperomyces virescens attacks ladybugs specifically at overwintering aggregations. The close physical contact of thousands of ladybugs in one location allows the fungus to spread from individual to individual. Infected ladybugs develop fungal growth on their bodies. The infection weakens them. An infected ladybug that survives winter will have reduced reproductive success the following spring. There is a second problem. The harlequin ladybug, Harmonia axaridis, introduced to Europe and North America as a pest control agent, has become invasive and is outcompeting native ladybug species, including your own. Harlequin ladybugs are larger, more aggressive, and carry a microsporidian parasite called microsporidium that is fatal to native ladybugs, but which the harlequins themselves carry without being affected. When harlequin ladybugs aggregate with native species at overwintering sites, which they do because they use similar sites, the parasite spreads. Native populations have declined significantly in regions where harlequins have become established. You find an overwintering site in late October, a south-facing wall behind the shed, where other ladybugs are already aggregating. You press into the group. The warmth of the aggregate is real. The temperature inside the cluster is measurably higher than the ambient air. You go dormant. Your metabolic rate drops. You're running on your fat reserves. Winter passes. Most of it is nothing, a kind of blank metabolic waiting. You are alive, but barely, breathing slowly, heart beating slowly, burning fat at a rate slow enough to make it through to spring. You emerge in March when the temperature rises above 10 degrees Celsius. You are thinner. Your fat reserves are lower than when you entered hibernation. The first aphid colonies of the year are not yet abundant. You spend several days eating pollen to supplement your diet while the aphid populations build. Then, in late April, you are walking along a stem when a parasitic wasp finds you. The wasp is Dinocampus coccinelli, a species that parasitizes ladybugs specifically. It stings you, lays a single egg inside your abdomen, and flies away. You continue walking. You do not know anything has happened. The wasp larva hatches inside you. It feeds on your internal fat reserves and avoids damaging your vital organs as long as possible, keeping you alive and functional as a host. Over the next 3 to 4 weeks, you continue your life apparently normally, walking, eating aphids, occasionally flying. Inside you, the larva is growing. Then, the larva exits through your abdomen and spins a cocoon between your legs. The cocoon anchors to the ground or a surface below you. You are still alive. You are positioned over the cocoon, like a bodyguard. Your legs twitch periodically, which repels predators that might threaten the cocoon. You are functioning as a protector of the thing that ate you. In some percentage of cases, the ladybug recovers after the wasp emerges. The survival rate for parasitized ladybugs is low, but not zero. If you recover, you will have reduced reproductive capacity. If you do not recover, you will have served as food, shelter, and bodyguard for something that never had any interest in whether you lived or died. It is late May. You are still positioned over the cocoon, twitching occasionally. The wasp pupa inside the cocoon is developing. In approximately a week, it will emerge and fly away. Whether you are alive to see that happen has not yet been determined. The aphids on the row stem below you are multiplying. No one is eating them right now.